All right, AP Bio students, let's talk about Unit 5 heredity. So I want to start from the beginning. There's a whole bunch of main points here. We're going to start with meiosis, so I'll tell you what the main stuff for meiosis is. This is a good point. A general familiarity with a ton of vocabulary. That is about right. Even if you don't know exactly what each word means, as long as you kind of have a vague idea of what it's about, you will probably be okay. Difference between meiosis and mitosis is super important. In terms of meiosis, crossing over and non-disjunction are probably the two most important things, kind of knowing when they could take place and what effect they're going to have. We saw that in the progress check FRQ question 2. It definitely showed up there. So yeah, that's a thing. Also, just like random recombination of chromosomes, also useful. The rest of this stuff kind of goes in other categories, so we can come back to that later. Recombination frequency calculations might be a thing, so we'll check into that later. But the rest of it comes in the next set of notes. So let's keep going. So meiosis. These are some words that actually apply to mitosis as well. Specifically, yeah, like all of these actually totally do. So we kind of talked about the difference between chromatin, chromosomes, and chromatids. Chromatin is just a description of DNA. It's the string of DNA with a whole bunch of proteins that kind of keep it bound and wrapped up. So when you see a cell, like the nucleus of a cell during G0, so during this normal phase, non-replication, non-meiosis, non-mitosis, just normal, DNA is all kind of unraveled. It's like spaghetti. It's all loose and it's all flopping around all over the place. That's so that transcription enzymes can make mRNAs. It has to be exposed. There's still an organizational structure. Protein, there's a whole bunch of structural proteins and other epigenetic tags. We'll talk more about epigenetics later. It's like, it means on top of genes, like epi, like on top of. They're all still there, but it's a lot more loose and open. And so that's the kind of chromatin. As soon as a cell is going to go into cellular division, mitosis or meiosis, it has to be kind of loose like that to replicate the chromosomes, to double the number of chromosomes. But after that, all the DNA gets packed into chromosomes. A chromosome is technically really just that big string of DNA. It's a single, not a single strand, but one string. It has two ends to it, which is the case in most eukaryotic organisms that have that. A lot of bacteria, just by the way, have their DNA is not in a string, it's actually in a circle. They're called plasmids. So most like bacterial DNA is like, it's in little circles. Not always all of it, and it might also be lots of those little circles, and it gets a little bit more complicated, but that's kind of a difference between eukaryotes and prokaryotes. But yeah, eukaryotes have these, and they have multiple sets of those. So each one of those is distinct, and it holds a specific set of information, like with specific genes that encode for specific proteins. Human beings have a total of 46 chromosomes in every single cell of their body. It's a good distinction there. Not just 46 somewhere lying around. No, every single cell of your body has 46, except gametes, except sex cells. And there's 2 copies of each. So you really have 23 types of chromosomes, but 2 copies of each, one from each parent. Sex cells or gamete cells only have half of those. They only have one of each type of homologous chromosome. Excuse me. Yeah. Chromatid is referring to when you have after the S phase of mitosis or the S phase of meiosis, either one, especially the first. I think there's only one S. Yeah, there's only one S phase for both. That's the synthesis phase. When you have the two, once the chromosomes have been copied, they're sort of connected at the centromere. So that's what the centromere is there. It's what holds the two together. Once that's happened, technically these are 2 different chromosomes, but sometimes they're referred to as one. like this whole thing is this is a chromosome, but this whole thing could be considered a chromosome. Technically, it's 2 copies of the same chromosome. In mitosis, they're identical. In meiosis, they might not be. They might have had some crossing over, which we'll get to later. But we call each of these as chromatids or sister chromatids. It's just kind of a way of talking about the fact that there's like 2 of them in this now chromosome. I don't know why they called this a chromosome and then this whole thing also a chromosome, but biology, man. Okay, so 2 sister chromatids and the centromere. So these are all useful things just to know, so that if you get confused about it, those are potential things to be confused about. As best as you can, it's good to try to keep those things straight and use the correct term for the correct scenario. But I also recognize that maybe that's not going to happen all the time, but you know, we'll just keep it in mind. Okay, so going on, we kind of mentioned some of these, but I'll run through them real quick with you. Somatic cells are all of your cells that aren't reproductive. Gamete cells are the ones that are reproductive. So typically ovum or spermatozoon or spermatozoa. So ova is plural, ovum is singular. I just love that that's the singular for spermatozoa. It's so weird. Anyway, homologous chromosomes I just mentioned. So this is a good example to help you see what homologous chromosomes are. So this is the haploids. So there's only, haploid just means like half of the normal number of chromosomes. So I always remember humans, I can actually never remember how many humans have. 23andMe helped though, because that reminds me, that's the haploid number. Yeah, 23, double it, 46. So your gamete cells will only have the haploid. They'll have 1 copy of each of the types of chromosomes. So they'll have, of the 23 chromosomes, they'll just have 1 of each of the types of chromosomes. And they are non-homologous, so they're not the same. Each one of these is different and encodes for different kinds of proteins and has different genes and different genetic information. A diploid has 2 of each type. So these are homologous, these 2. They're different from each other, potentially. Yeah, they are. They can't be identical because, well, definitely not in meiosis anyway. In mitosis, they would be identical. No, even then they wouldn't. That's not true because one's from mom and one's from dad. So there you go. Exactly. One's from the father versus the mother, maternal and paternal chromosomes. So they're 2 different chromosomes, but they just encode for the same information. So basically you have a double of all things, which is nice. It adds for some genetic diversity, and also if one chromosome has a really bad gene, the other one has one that is better, that can sometimes be helpful. Just depends. So yeah, there's 3 sets of homologous chromosomes. So that should help. That should make sense. Okay. Haploid and diploid. Yes. Lovely. Yes. Gamete cells have haploid, diploid. It's like half and double. Is kind of like how I remember it. It contains 2 sets of each one, 2N. So during the first stage of meiosis or mitosis, you end up with a double amount of that. Let me see if we can find a good this diagram has a good comparison of that. At least it's not terrible. So yeah, so for example, this is prophase 1. You end up with if the diploid number is this particular organism only has 2 chromosomes. So it has the short one and the long one, and it has 2 copies of each. So that's why there's 4. So during prophase 1 or prophase of mitosis or meiosis, you double all of them. At the end of mitosis, of course, you're back to 2N. And meiosis, you have half. So it's just another doubling. Or yeah, another doubling, yeah. So yeah, so that's why we use N. Diploid is like 2N. Yeah, haploid is like half of that, which is just 1N, then 4N. It's a whole thing. Sometimes it's kind of useful to think about, other times not so much, but hopefully it kind of makes sense. Daughter cells are copies of each cells. So that's the basics there. There's 2 other words that are useful to know. There's non-disjunction and polyploidy. And I don't know, oh, crossing over comes down later, good. Non-disjunction is when the homologous chromosomes don't separate. So they are supposed to separate here in anaphase, like this. But if they fail to do so, usually it's just like one. So like these two would still be together, potentially. This shows you some examples of like the types of mistakes that are possible. So if you end up with like half of the gamete cells, you know, have more chromosomes, have like 1.5 times the chromosomes, and basically there's two places where non-disjunction can happen. So here, these two chromosomes didn't split up. So one of the gamete cells only gets one of like the meiosis. During meiosis 1, you have one cell that's going to split further that has 2 chromosomes and one that has none. So it splits off. And yeah, this one, where would that one have gotten its genetic information from? Let's see what we got. Oh yeah, okay. So it would produce, sorry, it just took me a second. This is a cross right here, a breeding, or like, you know, the two mixing together, or a fertilization. So it's gonna, this particular development of this particular gamete cell is gonna produce two gametes that don't have this chromosome at all, and 2 of them that have double the amount they're supposed to have. So the offspring, if they are fertilized by a correct cell, you're going to find 3 copies or just 1 copy of that chromosome. In humans, this would definitely cause chromosomal disorders. Depending on which chromosome it is, it can cause different kinds of things. Yeah, we could have to look that up. I don't remember what some of those issues are. Sometimes it makes non-viable like embryos. So the embryos do not survive because having the extra chromosome or missing it is very bad. Other times it can just cause like developmental disorders of various sorts. There's never going to be no problems. Some of the problems might just be less significant than others. For example, if it happens with a sex cell, that's when you get into intersexed individuals who have different, which is one of the ways that one type of intersex anyway. That's a whole big topic. It's not all chromosomal, but chromosomal intersection is like XXY or XXX or XYY. There's a couple of different ones where you have too many extra sex chromosomes, which have various biological consequences, which some are much more serious than others. But we could look into that. That'd be a cool thing. That's something I used to know a lot about at one point, but yeah, it's cool stuff. Anyway, if the same non-disjunction happens, this is if it happens in meiosis 1, if it happens in meiosis 2, then you only end up with one cell that it's happening right here. 2 of the gametes will be perfectly normal, but then 2 of them will have issues. One of them will just have 2 chromosomes, 2 copies of the chromosome, and one will have none. So if it is fertilized by another cell that has the correct number, you'll only have 2 cells that have some sort of issues. All right, so non-disjunction is cool. This happens constantly in plants. Plants have a lot of non-disjunction and it doesn't actually cause them that many problems. And it ends up resulting in polyploidy. And polyploidy is when you have, instead of like diploid or haploid, you have multiple ploids like tetraploids or triploids or diploid. No, diploids we already have. Octaploids, hexaploids. In terms of viability, it seems to work much better if it's double the number of chromosomes rather than like, it's like in intervals of 2. Like having 3 of one chromosome doesn't seem to be as useful as having 4. There's kind of a diagram here that sort of explains how this happens. But what's kind of cool about plants is as long a plant, like if a human, if you have cells that don't have the same amount of chromosomes, and I guess this is true for all sexual cells, having the wrong number of chromosomes can cause issues in terms of creating viable offspring. But if you have like a tetraploid and another tetraploid, like a tetraploid egg cell and a tetraploid like sperm cell, they can actually produce like other tetraploid offspring, which is kind of cool. So that's kind of what happens with plants. That's a whole other area that you can look into. But especially like crop plants, especially grasses, tons of plants like strawberries are like polyploids, like corn and wheat and all those things all the time, constant polyploids. So if you have like a field of wheat, you could have like some of the wheat cells are like diploids, but some of them are like tetraploids or octoploids, and they all just kind of like can crossbreed with each other. I think it's partially because plants don't have to move. So like physically creating more like genetic information is less taxing on them. Unlike like for animals, it would be harder because it's like physically more mass, and that is like biologically not favorable. In terms of plants, it has benefits and downsides. Some of the benefits are those plants tend to be bigger, like the fruit. So like the wheat or the strawberries tend to be bigger. So in terms of farming, it's kind of a good thing. I think there are some issues. I didn't do a ton of research into this topic, but it is kind of cool just to think about the things that we would normally say like, oh, this is inherently bad, not necessarily are. So that's a whole nother area that you could look into, and maybe we'll get a chance, or I'll find some articles or something on it. Plants, polyploids, love it. Welcome to another episode of Mr. Apkin rambling about random science things that she half remembers. Here we go. All right, let's go into the process. So meiosis 1, it's really similar to just like mitosis. The only difference is that instead of the homologous chromosomes just kind of freely floating around and end up getting split during metaphase and anaphase, the homologous chromosomes actually like line up on top of each other and do this whole crossing over thing. They form what's called a tetrad because it has like 4 sister chromatids from the two duplicates from the parents. So it helps the crossing over helps mix the genetic information. This, when I printed it off, didn't show it very well, but this little crossing over shows that there are some different pieces. So they would say that this half was recombinant. So if you see the word recombinant is referring to crossing over because it's like recombining. So yeah, it's just switching genetic and switching genes. There's a whole thing about how recombination, how often it happens statistically, lets you map out chromosomes. We're going to talk about that later, but I'll show you the picture. Because if like 2 chromosomes or 2 genes are close to each other, they're more likely to recombine together, like at the same time. If they're farther away, they're less likely to. So it can help us to figure out where each gene is on a chromosome, like in relation to each other, which can be really helpful. Yeah, so 2 genes close to each other tend to recombine because they both kind of touch and split in the same way. But if they're far apart, it's less likely. Okay. So yeah, recombination is super important in terms of creating genetic diversity. This is why individual gametes will all have different genetic information. One of the reasons. The other one is also that when this diagram didn't print very well. Yeah, like just lining up on the metaphase line, it won't always be like paternal maternal. Some of them might be flipped over so that they get kind of recombined into creating gametes. So that's a big thing. Separation of homologous chromosomes is also another, I'm going to jot this in, source of genetic diversity. Because uneven split of maternal/paternal homologous chromosomes. See, these words are sort of important. Weird, right? Yeah, knowing what they all means. Okay, so yeah, that's the first phase. The second phase is just another split. This is when the sister chromatids split from each other. And again, it's kind of random. So in terms of crossing over and all that, which one gets which one is a big source of that difference. So If you're looking at this in the printout, like you can't see all the colors, which kind of sucks. But if you have it on your computer, overall, I think this diagram shows some of those crossing over events and the splitting of the meiosis one, splitting of the homologous chromosomes better. This one doesn't show it as well, but it does do a nice comparison. So, okay, so that is meiosis and that whole process. Knowing all of this is really important because that lets us have an idea of like how genes get separated. We're going to go over next is Mendelian and non-Mendelian genetics. What's kind of cool, it's kind of interesting to think about is when Mendel was experimenting with his peas, he didn't really know about like genes or chromosomes. That was not a thing at that time. So we're going to kind of mix and match all of that information. We could separate it into like what Mendel was able to figure out and then combine it with chromosomes. But like we know about chromosomes, we may as well just start kind of thinking of them together. But yeah, so that's that for right now. We'll come back and talk about Mendelian genetics next. We'll get into it a little bit. And I think I'm going to make 3 videos total. So this one, first part of genetic, of kind of inheritance. And then we'll do like the wrap up that kind of pulls everything together and adds some new cool stuff and goes back to chromosomes some more. So, oh, let me show you some videos that are related to this. My YouTube playlist for this unit is not very well organized, unfortunately. I'm working on it, but I'm having a hard time organizing it. But I do have, there's some videos right here all about just meiosis. This one is pretty cool. It talks about chromosomes and karyotypes. And it kind of goes through just some like misconceptions, like very common questions like FAQs. So that can be quite nice. A couple more meiosis ones. Some of these are really great animations that will kind of show you what it's like, what it looks like. This one is the one that's like in real life that I think I showed you, like actual, like microscope movies. Here's a comparison. The Amoeba Sisters are really solid. I like them. There's more of that. There's the rap battle I showed you. And then I don't think, yeah, the rest of this is other stuff. Oh, well, actually, this one is kind of useful. Yeah, let's pull this one up a little bit. Let's pull all 3 of these up and then we'll drag that one down. These 3 are all more about like chromatin. These 2 are about like chromatin and nucleosomes, like how DNA is actually packaged, which can be really useful to know. I'll probably throw those videos into Unit 6 as well, because I think they're relevant. Anyway, I think I'll stop talking there, and hopefully you got something out of that and it was vaguely interesting, and I'll see you in the next set of videos in the next video. Toodles.